Good afternoon, everyone. I'm going to present my work entitled Adenosine Receptors Differentiating Mediate Enteric Glial Cell Death Induced by Clostridioides difficile toxin A and B under the mentorship of Dr. Celia Warren from University of Virginia. Clostridioides difficile is an anaerobic bacteria that is the most common pathogen to cause healthcare-associated infections. C. difficile infection is increasingly recognized to be associated with high risk of functional diarrhea in patients after infection. In vitro, culture of intestinal mucosal layer and intestinal submucosal layer challenged with C. difficile toxin B resulted in much higher ILH than the culture with each layer alone. This find suggests that host components other than epithelial cells, such as enteric nervous system and immune cells, are also involved in the response to the toxins. The enteric nervous system is, is mainly composed of enteric neurons and enteric glial cells. Enteric glia can be found in the mucosal, submucosal, and myenteric plexus layers, and together with enteric neurons, enteric glial cells have been shown to regulate motility, secretion, and inflammation in other inflammatory bowel disease. Deletion of adenosine receptors is important in the pathogenesis of C. difficile infection. A2A being protective by decreased intestinal secretion, while A2B increases the pro-inflammatory response. There are 4 adenosine receptors reported so far: A1, A3, A2A, and A2B. Activation of A2A and A3 was shown to have an anti-inflammatory profile, while A2B has a pro-inflammatory profile. Most of the previous studies have focused on intestinal epithelial cells during C. difficile infection, but not on the components of the enteric nervous system. Our goal was to investigate the effects of toxin A and toxin B on expression of adenosine receptors in enteric glia and the role of A2A, A2B, and A3 on toxins-induced enteroglial cells death. Firstly, we investigate how C. difficile toxins modify the gene expression of adenosine receptors in a rat enteroglial cell line. We found that toxin A and toxin B upregulate A2A and A3, the anti-inflammatory receptors, and downregulate A2B, the pro-inflammatory receptors, in those enteroglial cells. Is also showing this immunofluorescence stain images for A2B. These adenosine receptors were involved in enteroglial cell death induced by C. difficile toxins. For that, we used a pharmacologic approach by administering specific adenosine receptors agonists and antagonists. Let's show here activation of A2A with ATL313 and A3 with a chloride IB nickel decreased enteroglial cells death induced by C. difficile toxins. And we found that blockage of A2B receptor with ATLH01 also decreased enteroglial cell death induced by toxin A and toxin B. Given that in vivo enteroglial cells interact with other cells, we assessed whether A2A and A2B deletion would prevent enteroglial cells death during infection. Use this mouse CDI model. We euthanasize the mice on day 3 post-infection, which is the peak of infection. And we found that infected A2A knockout mice had high percentages of apoptotic enteroglial cells, while infected HB knockout mice exhibit low percentage of S100B positive to non-positive cells compared to wild type infected mice. The changes in adenosine receptors in enteroglial cells caused by toxins may be one of the factors involved in modifying the severity of diarrhea during Clostridioides difficile infection. In conclusion, all finds showed that C. difficile toxins downregulate H2B receptor and upregulate H2A and H3A enteroglial cells. These receptors appear to play a regulatory role in enteroglial cells death induced by toxin A and toxin B, and modulation of these receptors may protect enteroglial cells from the deleterious effects of CDI in the enteric nervous system. I would like to thank the support of everyone in Dr. Warren's lab and Dr. Brito's lab. Thanks also to the organizers of CrossPath for the opportunity to present our work. Thank you.